Hi, my name is Moran Frenkel-Pinter, and I'm a research scientist at Georgia Institute of Technology. I am a biochemist and a member of the NASA Center for Origins of Life and the Center for Chemical Evolution. And today I will tell you about the current view on what we know about chemical evolution and how we can peer into the past, as Ram Krishnamurthy said. If you have any questions or comments, please feel free to email me to moranfb@gattech.edu. So let's start. The outline for today's primary talk is the following. We will first define chemical evolution. We will talk about the elements of chemical evolution and how did chemical evolution invent Darwinian evolution. We will connect geochemical cycling with chemical evolution. We'll talk about some promising future avenues and summarize the talk. I want to start first here by defining chemical evolution. So the literal interpretation of chemical evolution is change over time, as in evolution, of chemicals. And in the broader sense, really, chemical evolution is the process that describes chemical changes on the primitive Earth that gave rise to the first functional polymers. We heard in the last primary talks about the buildup of that chemical complexity on the prebiotic Earth, and the process that acted upon the chemical inventory and gave rise to higher orders of emergence, such as structure and function, is really chemical evolution. And chemical evolution had different stages, and I'm representing here some of them. So first of all, we had the starting inventory, monomers or building blocks that reacted to form short polymers, which we call oligomers. We probably had both homogeneous polymers, or oligomers, so from the same kind, and heterogeneous polymers, having different kinds of building blocks. And oligomers were linked together with each other and with monomers or building blocks to form longer and longer polymers. And there is a refinement in polymers with an increased functionality that really laid a foundation for extant polymers. When polymers arose, we also had the emergence of things like structure and function. And eventually we got homochiral polymers. And when we got to things like informational polymers, then really, such as protonucleic acids, it's a whole new game. But what I will discuss here in this talk is what happened before we got to informational polymers. And the chemicals that we have in life today came from this process of chemical evolution. Once life arose on Earth, they operated by a set of rules that we can track very nicely throughout biological evolution, of which we have really a vast recorded history that we can look at, from fossils to our own DNA across different biological species. And in biological evolution and Darwin's theory of evolution, open-ended chain over successive generations is really based on natural selection, which operates based on heredity, variation, and selection. So we needed informational polymers such as nucleic acids, but what happened before that? So we must have had if this is Darwinism in biology, we must have had a pre-Darwinism, like a chemical evolution that led to the first informational polymers. And the question is, how do we look at chemical evolution? So most of the history is now gone, and we only see the end results. And we have a pretty good idea on the starting point in terms of what is the molecular inventory. We know it from meteorites and from model prebiotic reactions. But what happened here in between? This is the question. And generally speaking, we can look at this question from both, so pre-Darwinism, that's what we're talking about. And we can look at this question from a bottom-up approach, where we look at the starting inventory and build up complexity from there, or we can look at it from a top-down approach, where we look at what is in extant biochemistry and get close to what happened before, as Ram Krishnamurthy mentioned in his talk. And, as I mentioned, I'm not asking what happened after we got informational polymers, but really what happened before. And this collective process that I'm showing here is the process of chemical evolution. And in the context of chemical evolution, I really love that concept paper by left French biologist and Nobel laureate François Jacob, who talked in this science paper in 1977 on evolution operating as a tinkerer rather than like an engineer. And he pointed out, all these years ago, that the really creative part of biochemistry must have occurred really early on. So that means before life arose, during chemical evolution. We know that today, the biopolymers of life, our biochemistry, the metabolism, they're very, very conserved. So how did we get to the extent toolbox of life? Through chemical evolution. And when we talk about the tree of life, the estimates are that more than 99% of all species that ever lived on Earth have died out. So we only see 1%, and we don't see 99% of all life that existed on Earth. And this is the tree of life. This is the time axis on the left. And we see the green branches, which they represent the survivors, and the white ones represent the species that are now extinct. And the process of evolution that really preceded that life, the first life on Earth, is the chemical evolution. And here we see Luca, the last common universal ancestor, which is the most recent common ancestor of all current life on Earth. But what if I'll tell you that what we see here in the tree of life really resembles the process or the evolution of molecules? And if we'll draw a very similar tree, this is what we can think of. And again, so we'll see the polymers of life, the species that survived as the green branches, and the ones that did not as white branches. So the polymers of life, we know we have polypeptides, we have nucleic acids, so we have two types, RNA and DNA. We have a variety of sugars and of lipids. And we can ask, how much species are still exist today? So it's I cannot tell you the exact number, but it's a very minor portion of the possible polymers that have existed. And we only see the survivors. And on top of that, on the tree that I showed you, there is all this chemical connectivity in extent biochemistry between the biopolymers and monomers at different levels, directly through chemical linkages and indirectly, for instance, by co-assembly. As an example, I will show you chemical interconnectivity of amino acids, which are the building blocks of protein. And we know that in addition to incorporating into proteins, we know that in the ribosome, amino acids are linked through an intermediate in which they are linked to tRNA, which is an RNA, in the process of them incorporating into peptides in the ribosome. But there are many other examples of copolymers and conjugates. So here we see that many glycoproteins and proteoglycans are actually linking proteins and glycans together. And we know that glycans that are covalently linked to proteins comprise about 50% of more of the total molecular weights of glycoproteins. And acetyl-CoA here is a major player in metabolism and contains amino acids, phosphates, adenine, which is a nucleobase. And we have lipopeptides and peptidoglycans. A lipopeptide is a molecule that has a lipid connected to a peptide. So really, the polymers are maybe synthesized, as pure, but then they undergo all of these modifications and post-translational modifications and become heterogeneous. So the biopolymers of life are not chemically distinct and are intermingled in so many ways. Now, I want to talk about how did we get to evolved polymers and molecular networks. So what are the elements of chemical evolution that we have in mind? So first, we'll have different forms of chemical selection that operated on multiple levels. Polymerization. Polymers will do things that monomers cannot, so we need to link the building blocks together. And we've heard about it from Luke Lehman. We also want to have reversible reactions and to explore the chemical space. So kinetically accessible near equilibrium chemistry will be very beneficial here. Another element would be environmental cycling, so oscillating reaction functionalities, directionalities. Open-ended change. So if we have equilibrium or steady state, it really equals fail. We need a process in which there is this possibility for and indefinite increase in complexity. And open-ended evolutionary change needs a dynamical behavior that will allow complexity to grow in an unbounded way. And here we'll have molecular memory, pre-genetic before Darwinism. Memory itself had to undergo some form of evolution and probably started with more primordial and simple forms. And we also need some form of energy harvesting. So we need to push the reactions forward, and to do that, we need some energy. And there's a broad community of scientists that work on these different elements of chemical evolution, and I'm listing some of the groups here. So of course, we have NASA's PCA3 network, we have the Center for Chemical Evolution, the NASA Center for Origins of Life, or COOL. We have the Earth Life Science Institute in Japan, the Origin Center in the Netherlands, the Cronin Group, the Baum Group, and more. And all of these elements are experimentally accessible and can be validated experimentally. So now let's break down some of these elements. Let's start with chemical selection. And there are different selection forces that could lead to selection of building blocks. So first, the inventory. We cannot select for something that is simply not there. Chemical reactivity, co-assembly, function, and there might be more levels than that. So chemical selection operates on multiple levels. So imagine that very first step of linking different building blocks or monomers together. We know that some will work better than others. For instance, in the example that I showed you, you can see that if we look at the first linkage of different monomers into oligomers, you can note that the squares do not form linkages with other molecules, so they will be excluded. And there are different experimental demonstrations of this. So for instance, Williams, Hudd, and Lehman groups showed that there is a chemical basis for selection of today's cationic amino acids over non-proteinaceous analogs due to their intrinsic chemical reactivity. And Andrew Brock also showed some selective polymerization of certain amino acids, mostly proteinaceous, that we have in today's biology. So there are different examples of that. Looking at extant biochemistry, it is very clear that the building blocks of life were selected based on water activity. When we think of proteins and their building blocks amino acids, when we link these together to form the peptide bond, we lose water. Same with carbohydrates. When we take two sugars, we link them together, we lose a water molecule. Also with nucleic acids, having a sugar and a base connected will lead to loss of a water molecule. And the same when we attach the phosphate. And it's very similar to lipids. So it is very clear in today's biology the importance of having a water-based chemistry. And this is the second level of selection, assembly. The things that survived were selected. And if there's just one takeaway message that I want to remember from this primary talk, is that structures or co-structures will lead to slower degradation rates. Imagine having these two different polymers with different backbones. And one polymer can fold, let's say this one, and the other one cannot. So now we'll put them in water, and the polymer on the right will now fold. And this here, really in water, we will see that the equilibrium is towards degradation of these polymers, so towards hydrolysis. And this, having this different folding ability or assembly, will lead to differential rates of degradation. That polymer that folds will not degrade, whereas the one that does not will. So this process will lead to enrichment of species, such as this one, that persist. Ribosomal RNA, for instance, degrades very slowly. Cellulose, which is a polymer of glucose, is basically indestructible. Amyloids involved in a variety of neurodegenerative diseases and other protein misfolding diseases are very resilient. Same for spider webs. So it is a general phenomenon of biological polymers. And chemical selection operates on the basis of assembly. Things that assemble will persist. This can also explain the emergence of homochirality. So homochiral polymers tend to be more stable than heterochiral ones and will degrade more slowly. So this is a very strong selection force favoring homochirality. And we can see emergence of structures very easily in simple reactions. Here on the left is work from Jim Cleave's group showing formation of microdroplets after heating a mixture of different hydroxy acids together. And here on the right is work from Jason Greenwald showing that volcanic gas carbonyl sulfide led to condensation of amino acids into peptides that form these tertiary structures that resemble amyloids that we see in today's biology. And there is a known platform for chemical evolution, which is called wet-dry cycling, in which we'll see shuffling that occurs at both phases of the dry-wet cycling. So in this scheme, we see that we had conditions where it's really dried and hot. Under these conditions, we will favor spontaneous condensation between the different building blocks to link them into polymers. Now imagine having a wet phase where rain occurs, and we'll have these polymers now in water. And as I mentioned, some will form stable structures, some will not. Some will assemble with other species, some will not. And this will lead to differential rates of degradation of different polymers. We'll have polymers that persist and polymers that do not. The ones that do not will replenish the soup, will degrade and replenish the soup with fresh materials. And you can imagine doing this cycle over and over, so doing dry-wet cycling, which eventually will lead to more resilient polymers following evolution. So we talked about the different elements of evolution, and we see them here. We see the chemical selection on multiple levels. We see polymerization, exploration of chemical space, and environmental cycling. And there are many groups working on this. The researchers that really push this idea forward are, for instance, Nick Hudd, David Dimmer, Lee Cronin, but there was a theoretical work by Michael New, and there are many others. And we already saw this example, which is really chemical evolution. We have mixtures of hydroxy acids that react and polymerize. So this is chemical, there is chemical selection we talked about of building blocks based on chemical reactivity. And this is an example of chemical selection that operates on building blocks. And we see that we'll have these hydroxy amino acids linked together, and we will have the backbone itself showing evolution, because first we will have a lot of esters and eventually they will evolve more towards peptides. And dry-wet cycling is a geochemical energy flux that can drive prebiotic chemical processes. Dry-wet cycling provides a direct link between biochemistry, so we degrade polymers by hydrolysis and we condense them to link them together, and the environment itself, dry-wet cycling. Water activity on land surfaces cycles with the rotation of the Earth itself. And one very easy way to get to dry-wet cycling is having a spinning planet with land. For instance, imagine this beach in Hawaii, but it can be a desert, and it could be many other locations where we have small ponds or small surfaces that can undergo this dry-wet cycling or cycling in water activity. And to drive chemical reactions, we need energy. This was one of the elements. And all these sources of energy could have contributed to chemical evolution probably. So thermal vents, sun impacts, electrical energy, so lightning. And what were the postulated locations for chemical evolution? I mentioned land, and there's really no need for big surface areas, just also small ones can do. Hydrothermal springs, oceans, so these thermal vents. Air, can we have chemical evolutions in aerosols? After we formed polymers, so we've talked about all of these different elements of chemical evolution. What I want to mention here is that after we form the polymers, we can very easily get to functional polymers. And here are some examples. This is in this work by Irina Mamezhanov. She showed that hyperbranch polymers possess high photocatalytic activity. And Lee Cronin explored the hypothesis that disruptive equilibrium via recursive cycling imposes selection forces and showed the emergence of catalysis, specifically catalyzing what's called ester hydrolysis. In this work by Williams, Lehman, and Hudd groups, we showed the emergence of protopeptides that stabilize nucleic acids. So the really creative power of chemical evolution gives us many possible functional polymers. And to end this part of the talk, here are the survivors of today's biology that we have in life. This is the toolbox of life. Our biopolymers, the main ones, we have protein, sugars, nucleic acids, lipids, and we have many other molecules that are found not in the polymers, but as small molecules and metabolites. For instance, the hydroxy acids that we talked about are found in biology as metabolites. And if the conditions were different on the pre-biotic Earth, it is possible that we would have had other survivors. But these are the ones that we have today. Now I will discuss some of the promising future avenues in the field of chemical evolution in my mind. One such direction is to look at things not from a top-down approach where we know exactly what to look for, but explore the huge chemical inventory using a variety of building blocks. Nick Hutt is looking at the chemical space of plausible prebiotic nucleobases that are not found in today's biology and possess some very interesting properties. Ram Krishnamurthy showed us that considering mixed nucleic acid backbones, they show that chimeric templates can give rise to homogeneous backbone sequences. And as another example, in the case of mixtures of hydroxy and amino acids, which we've heard about, we know that they can eventually evolve the system towards peptides, even though the starting material was using hydroxy acids, which are not found in peptides today. Systems chemistry is another interesting direction in my mind, and it is emerging as a promising direction and a relatively new subdiscipline of chemistry. It is the study of networks of interacting molecules that give rise to emergent properties such as structure and function. And in the context of origins of life, there is systems chemistry investigation of the emergence of both the chemical inventory itself of small molecules and the emergence of polymers. And what about complex mixtures? So how can we understand the emergence of complex phenomena when having complex starting inventory? This is one of the questions asked in the Baum lab, and they looked at mineral-associated mutually catalytic systems rising from complex mixtures. Molecular cooperation is another very important concept in my mind, which is being promoted a lot by Lauren Williams and is my specific subfield. We know that all known extant biology relies on connected networks of proteins, nucleic acids, carbohydrates, lipids, and small molecules. And it is very likely that biomolecules co-evolve from the very, very beginning. We know that in today's biology, RNA makes proteins in the ribosome, and protein makes RNA in RNA polymerases. And this interdependence of biology leads to a paradox for the start of life. Which came first, nucleic acids or proteins, the chicken or the egg? And coevolution models suggest that neither nucleic acids nor protein came first, but rather that RNA and protein were selected together through a process of coevolution. So the idea here is that just as we have mutualism in biology, in which we mean interactions in which both species, as in organisms, benefit, for instance, the bee pollinates the flower and the flower supplies food for the bee, that it's the same thing with chemistry when we talk about mutualism in chemistry, where it is an interaction in which both species, as in molecules, benefit. And here I'm showing you one example, which is the ribosome, the oldest molecular fossil, which is a machinery made out of RNA and protein. So how far ago these types of mutually beneficial interactions occurred? And I would like to point out some burning open questions to the community before we end this primer talk. What was the chemical inventory? How did it vary over space and time? What were the conditions on Earth and how did they fluctuate and how much? Land in terms of temperature, atmosphere, redox state, and so on. How did impactors affect the conditions on Earth, bodies of water, inventory? What did thermal vents deliver? What did asteroids deliver and what did atmospheric chemistry deliver? Can dehydration chemistry happen during freeze-thaw cycling or on surfaces or subcompartments in aqueous media? So there are many open questions. And the summary of this primer talk is that chemical evolution is really the process that describes chemical changes that gave rise to the first functional polymers. And chemical evolution has several very basic elements. We talked about chemical selection on multiple levels, polymerization, exploration of chemical space, environmental cycling, open-ended change, evolution, the actual evolution of molecular memory, and energy harvesting. Chemical evolution can be studied both theoretically and experimentally, and molecular memory and chemical complexity are not well understood. And I want to end by saying that while we may not be able to recapitulate life, we can hope to understand some of the chemical principles that led to it. I hope that you enjoyed, and this sums up my talk.